it looks like this. These leaves have been inoculated with agrobacteria. And we have green fluorescent protein. And this picture is not lying. It's really bright. It produces a lot. Now I have a vector that produces, it's one of the top 3 vectors in the world. It's now patented here at Baylor University, got a patent for it. And it produces prodigious amounts of protein, of green fluorescent protein. It produces so much that I can put a UV light against the plants. We have the room lights off, and you can see, it's bright. You put the room lights on, you can still see it. It is incredibly bright. I can produce 40% of the total soluble protein in the cell is my GFP with my vector. So it's just a really powerful way to produce it. And I bring this up because it's plant virus vectors, but you can use animal virus vectors to also deliver all sorts of things. So I wanted to get you into viral vectors now, because when we talk about vaccines, you can also deliver vaccines into humans this way, and especially into for veterinary vaccines as well. Okay, so those are plant viral vectors for protein expression. Any questions before we leave that? Okay, our next one we're going to look at then is fusion partners, and a better name for that, it's a better search term for that is affinity tags. Okay. Somehow I got into cap locks, but anyway, affinity tags, and we're going to go to, for this one, we're going to go to the wiki search for this, protein tag, Wikipedia. And what protein tags do, we don't have a picture for it so much. Well, let's go back here, actually, to these images so we can give you a picture of this. Protein tags let you do all sorts of stuff. Here's one. Yeah, this gets us into affinity chromatography, which we'll get into next. But yeah, here's a good one. These are flag tags. So flag is a type of protein tag. We have our target protein, and then you add the flag peptide, which is just a aspartate, a tyrosine, lysine, aspartate, 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 lysine, D-Y-K-D-D-D-D-K. You just include that in your in the genetic engineering of your target protein. So you're doing PCR to amplify the gene for your target protein. Slap in a flag tag in there while you're doing the PCR, and you create a gene which has your target protein, but it has a little tag on it also. Well, they have an antibody, the flag antibody, which really recognizes the tag really well. And so that's why we have it. And look at that. I mean, you've got aspartates and lysines and tyrosine. These are all, well, tyrosine's nonpolar, but the aspartates and the lysines are really very charged, right? And so they are easily, they have a big presence. They're not like a bunch of little glycines and alanines laying around. These have a real distinctive signature to them. So you can design an antibody that binds to them very well. And they sell this flag antibody. Anyone can make the tag. It's just a matter of how you engineer it. But the antibody you have to buy from Sigma that sells these. So what you can do then is you have your target protein, and let's say you don't have an antibody to your target protein, you're expressing it in a system. You can see if your system is producing the target protein by simply doing a Western blot, and you use the flag antibody. So no need to go to the trouble of making an antibody against your target protein, which is a lot of work and expense. Instead, you just add a flag tag, you buy the flag antibody, and you can visualize it on Western blot easily, or ELISA, or anything else you want to do. Now, the other great thing about this is you notice they have this flag antibody coupled to a Cephalose bead. What you can do then is you can go ahead and make a column of Cephalose, which has the flag antibody attached to it. And Sigma will gladly sell you that. Then you take your target protein from a cellular lysis, just a crude mix. You run it through the column, and then you wash, 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 and your target protein is stuck to the Cephalose column. And then you can use special chemicals to get this off over there, and you have the target protein beautifully purified. So that's why, that's another good thing about affinity tags, about fusion partners. There's a third good thing to affinity tags, fusion proteins. And this one doesn't do it, but if you have a bigger protein on there, let's say you have a protein that might be 30 kilodaltons or something, then if you have a target protein which is naturally unstable, it tends to fall apart. And you'd say, why in the world would we have unstable proteins? When you're doing something important in the cell, you're generally going to be unstable, because you don't want a protein to be stable and stay in the cell too long. Because if you have an important thing done in the cell, like apoptosis, too long, then you could kill the cell. So many of the really cool proteins we want to look at are unstable. When you fuse them to the fusion partner, they magically become stable. When you fuse them to GFP or GST or maltose binding protein, and we'll look at those all in turn, they become stable. So three things that affinity tags are great for. One, you can identify them with a single antibody. Two, you can use them for purification. They're a purification tag. Three, they stabilize the protein. Those are all great things. So we'll go back to the, I'll look at this here. This just gives you a picture of affinity chromatography, which we're going to get into in a moment. Well, we may as well talk about it now. We're already let the cat out of the bag. Affinity chromatography is you can bind all sorts of proteins on here, but they just happen to stick to one element on the Cephalros beads. And so it could be, for example, an antibody, which would be a classic case of affinity chromatography. And then you wash, 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 and get rid of all the other proteins, but your one protein which sticks to the affinity tag is sticking, and then you elute with a particular type of chemical, and out it comes. And then you want to get rid of that chemical maybe, and you could send it through a desalting column. Okay, and that's depending, that's an exclusion column, which we're not going to get into now, but it's just how well they stick to the beads. Okay, so that's affinity chromatography. Let's go then to the web. And we're going to look at protein tags for Wikipedia. And when do we go to? To 9:30. Yeah. And here we're going to have in this case we have a lot of different ones. We have maltose binding protein, chitin binding protein, glutathione S-transferase. These are all really big affinity tags. And you can look at these if you want to. And there are also other ones like the flag tag right here. And there's the polyhis tag, or commonly called the 6-his tag. And this is 6 histidines in a row. So it's like the flag tag, except it's 6 histidines in a row. And they're all used basically the same way. So that's all we have to go through on this one. There are MIG tags and HA tags, and that's actually from a virus. They've got a really good antibody to that HA, that hemagglutinin. Okay, so let's go ahead and come to site-directed mutagenesis. And I'm going to just cover one thing in site-directed mutagenesis, and that's this. We're going to cover overlap mutagenesis. And I'm going to look at some pictures here. I think this should do it for us. Overlap mutagenesis. So for overlap mutagenesis, we have 2 PCRs which are done. Yep, showing up pretty well. Okay. Now that says 2:28 on it. I hope. Did we talk 2 hours? No. Oh no. Did it start and stop? I hope it's recorded it. Yeah, it was blinking all along, wasn't it?
Yes, been blinking.
But now it says 2:42 on it. I don't know if it. hope that worked. Okay. So for here, what we have is we're going to have a PCR1, which occurs, and it's right here. And then we have a PCR2 right here. And we have one set of primers. There's one here, and then there's one here. And then we have another set of primers, one here and one here. You notice that these two primers are overlapping. And you notice that we have a cross right here. The cross represents the mutation, the plus sign here. And so you notice both of these have a mutation and they're overlapping. So by doing PCR1, we now have this one PCR here, and it has the mutation right here. PCR2, we have the mutation here, and we have the mutation right here. I guess we could call this PCR1A and PCR1B. Let's call it that, PCR1A, PCR1B. Now, we have, we take these two PCRs, and you notice they are indeed overlapping. So if you run them through a thermocycler, you'll see that when they overlap, here's the free 3' end, they serve as primers for each other. And you can extend this and extend that. And you get the whole piece that you originally started with the mutation now included, right where you wanted it to be. In actual fact, you usually do this reaction for only 5 cycles. And then you take primer A and D and pop them back in. You see how you have primer A and D here. So you do 5 cycles of this stuff just to get the full template, and then you add primer A and D, and it's much more efficient at that point, and then we get the whole piece put in here. Then you can insert it back into the vector where it came from, and you have the mutation where you want it. So that's overlap mutagenesis. It's a type of site-directed mutagenesis. Overlap mutagenesis can also work this way. We have 2 completely different sites. Let's say this site is a signal peptide sequence, and this site is a gene. And you want to add the signal peptide to the upstream of the gene. So what you do is you would have an A primer here, a B primer there, and this B primer recognizes the C terminus of the end of the open reading frame for the signal peptide. But we add a little tail on here, which is just an overlap. Your choice is an arbitrary sequence. We add the complement of that arbitrary sequence here in primer C. And then this recognizes our gene of interest. We do PCR A, PCR B, we get the signal peptide and the gene of interest with some arbitrary overlap, but the overlap is identical here. Then, just like we did before, we denature, put it in a thermocycler, and these, at this arbitrary overlap we've created, we have, this serves as a primer for this, that's a primer for that. Use A and D to finish the job off. And now we have a chimeric piece. Here's our signal peptide, and then there's a little bit of arbitrary overlap. And then here's our gene. And we can pop it back in here. And just like that, we've stitched a signal peptide onto our gene. So that's site-directed mutagenesis. Now, our final one here is random mutagenesis. And for random mutagenesis, we have, let's see, as I recall, 8 approaches to random mutagenesis. That's right. Yeah. So here are 8 approaches to random mutagenesis. What random mutagenesis does for you is it has random mutagenesis allows you to insert all sorts of different mutations. And so let's say we have a gene that you want to mutate. Then with that gene, you can with that gene, you can with this gene, you are able to introduce all sorts of different mutations. All sorts of different mutations occur. And so now we have you can look at what you have this gene, you have all sorts of random mutations in it. You are able to test all those randomly mutated genes and see which one just happens to have a better, more activity or a different activity or whatever it may be. So this is all random. It is, you are not planning it per se. You just want to look at hundreds of random mutants and see which one might have better activity or a different activity, the one you're looking for. So here are some different ways. One is error-prone PCR. If you increase the magnesium chloride concentration in your PCR, you will get more and more mutations the more you increase that. It just tends to increase the error rate of the PCR. PCR already is pretty error-prone, but you will increase that rate. So if you use regular standard Taq polymerase and increase the magnesium chloride concentration, you will get errors in your PCR. Then you can clone that back in and see if you have, just randomly, you've created new genes which have errors. Another way is you can use mutator strains. So Stratagene company sells this XL1 red, and when you mutate things in this strain, the genes tend to accrue a lot of mutations. Another one is you can have random inserts occur. So there are kits which will randomly insert 15 base sequence throughout a whole protein, and it will induce mutations that way. By instead of single base mutations, it will give you 15 base insertions. And then we can finally go to the chemicals. So EMS, or nitrous acid, are chemical mutagens. So you simply treat your DNA with this, or treat your cells with this, and they will, not this DNA, but you treat the cells with this, and the cells will mutate. You have to be very careful because you don't want to expose yourself to these mutagens because you can get cancer. And finally, we have DNA shuffling, which we're not going to get into now, but this is a means of inducing all sorts of changes. What it does is it takes different pieces of the gene and it shuffles them randomly. It's a complicated process, but it basically takes all the different pieces of your DNA and it randomly shuffles them all and you take pieces of DNA from all sorts of different organisms, and it maintains the order of the DNA. As long as let's say we have a gene, it's an esterase, and you want to get a new type of esterase. And you take this esterase from all sorts of different organisms. It will keep the order of the esterase, but it shuffles the different pieces between different species. So let's say we have 10 different esterases from 10 different species. You do DNA shuffling. The order will be maintained, but the first part might be from one species, the second part from another species, the third part from another species, and you create new genes this way, which have new properties. So that's random mutagenesis, and I think we're finished with that.